my name is Claire. I'm a junior year mechanical engineering student in University of Michigan. This summer, I was working with Professor Arpo in his Human Biomechanics and Control Lab. I have been mostly working with my research topic, the energetics of perturbed standing. Besides, I've also helped around with other studies, such as IMU gate variability test, complex terrain study, and encoder scaling factor calculation. Today, I would like to briefly introduce and talk about the perturbation project I have done in the last few months.
The purpose of this research is the study of mechanics and energetics of human movement. Energetics refers to the metabolic or chemical energy expended by humans to produce movement. Mechanics refers to the importance of the dynamics of the human body in nearly all movements. In this earlier stage perturbation project, we were interested to see how much metabolic energy change with different levels of perturbation, how much work done by the joints during the perturbed activity, the center of mass work rate, and all power consumed. In addition to the perturbed experiment, we would also like to find out the non-perturbation joint angle correlations. The metabolic energy may be estimated through measurements of oxygen consumption. Lab techniques for studying mechanics including analytical models, computational simulations, and recordings of kinematics and forces produced during movements. There were 5 subjects participated in this study, including 3 males and 2 females. Vicon motion capture system, oxycon, force plate, and low-cell stick were used for experiment. Lab data were processed through Vicon Nexus, Visual 3D, and MATLAB. There are 2 parts in this experiment, perturbation and non-perturbation test. With perturbation test, subject stands normally on 2 overground force plate with oxycon devices. Low-cell stick was used to apply poking forces to the subject in every 5 seconds. 4 trials have been conducted and each lasts for 6 minutes. With non-perturbation test, subject stands normally without any external forces. 2 conditions have been conducted and each lasts for 10 minutes. For all experiments, Vicon captured all markers data. The sampling rate for Vicon is 100 Hz. After running static and dynamic calibration, all Vicon data were initially processed and saved in C3D format. The first analysis for this study was metabolic energy consumption. We collected 6 minutes data and only used the last 2 because the carbon dioxide and oxygen flow rate will tend to be stable during the last 2 minutes. We calculated the work rate done by a default function and normalized by subject mass and leg length. We picked 3 male subjects' data for analysis, and the metabolic energy consumed by poking forces were found to increase about 20 to 30% compared to the normal stance. We also analyzed the joint angle, moment, and power done by subjects. We used the last 90-second data for analysis. All data were calculated by model-based computational pipeline. We run 6 Hz low-pass filter for all motion and 25 Hz for force data. When joint angles were calculated, we found the peaks on MATLAB, stretched and shortened each cycle to 1,000 frames, and averaged them to get a single cycle. Later on, we're able to get the 3x3 plot for joint analysis, which shows the angle, moment, and power data on ankle, knee, and hip. The reason why the power plot fluctuates about 0 is that the subject first did negative work to resist the perturbation forces, and when the force removed, subject did positive work to get back to the normal position. In this situation, hip did most of the work and ankle did the least. Another thing we were interested to see is the center of mass work rate change during perturbation. Again, we collected the last 90-second data and did the same low-pass filtering. We added all force components with reference to the lab and subtract the gravity. Integrate forces over each poking activity, we were able to get the center of mass velocity. The boundary condition would be 0 velocity during normal standing. Integrate power over time, we were able to figure out the work done by load cell, as well as ground reaction force. There is a slightly offset between the positive work done by load cell and negative work done by ground reaction force. The reason would be a time delay caused by human reaction. Theoretically, the sum of all positive work and negative work should be equal, and the power returns back to zero when perturbation removes. Lastly, we would also like to see how each joint correlated by conducting the non-perturbation tests. We collected 6-minute normal standing data and processed with 1 Hz high-pass filter. Joint angles were found by running Visual 3D pipeline, and then we differentiate the angles to get angular velocity and acceleration. Note that there were a slightly offset between the Visual 3D result and MATLAB result. They might caused by different methodology or coordinate system correlation. In order for us to understand the joint correlations, we plotted every tube joints and calculated their covariance. After high-pass filtering, the shapes of each tube joint were similar to ellipse shape. More narrow the shape is, more strong the joints were correlated. The results found from this study indicate that the knee and ankle joint has the strongest correlations. Note that here I define joint extension to be positive. Due to the technical limitation, there were a few shortages of this lab design. We applied perturbation manually, thus the forces were not consistent and well controlled. Because of it, the third variables like the time slot and the poking positions were also hard to control. Therefore, a well-designed control system is needed for accuracy and reliability. In the following semester, I will be continuing my research with perturbation devices. Motor, amplifier, load cell circuit, and microcontroller will be added to the experiment. Further, we will conduct walking perturbation trials as well as standing trials and collect about 8 to 10 subjects. Lastly, I pretty enjoyed the work I have done in this summer and hope I can get more exciting findings from my future research studies. Thanks for watching.